So now we're going to begin talking about incomplete dominance and codominance. Incomplete dominance is when the heterozygous individual is a blend of both homozygous parents. I want you to go ahead and write this down on your worksheet. For example, if we have a red flower and we cross it with a white flower, the offspring is going to be a blend of both, which is pink. We're going to go ahead and work this out on Punnett squares. If we go ahead and start our Punnett squares crossing the red and the white flowers, we notice that in the F1 generation, 100% of the time we're going to have pink flowers. When you go ahead and cross both of those pink flowers, in the F2 generation, 25% of the time you're going to have big R, which is going to be red. 25% of the time it's going to be white flower, and then 50% of the time you are going to have pink flowers. Codominance is when both of the parents' traits appear together in the offspring. I want you to write this definition down on your worksheet. For example, if we have a blue fish and we cross it with a red fish, the offspring are going to be a combination of both blue and red. I'm going to show this example using Punnett squares. In the F1 generation, notice that 100% of the time we're going to have a fish that is both a combination of blue and red. An example of a codominant trait is going to be red blood cells. We have 4 different types of blood: A, B, AB, and O. The genotypes are going to be written with a capital I. This stands for immunoglobulin, while there's a superscript for either A or B, which is going to represent the blood type. And here we have a little I, which is a recessive trait for O. So for blood type A, it's either going to be written as AA or AI. Blood type B, written as BB or BI. Blood type AB is written with an A and a B. And blood type O is little I. So blood type A is going to have an A antigen. Blood type B is going to have a B antigen. Blood type AB is going to have both A and B antigens. And blood type O is going to have 0 antigens. An antigen is a protein on the red blood cell that detects anything that is foreign. Go ahead and write this down on your worksheet. For example, if we have a red blood cell that has an A antigen and it's surrounded by other red blood cells that have the A antigen, everything's going to be just fine. But if we were to introduce red blood cells that have the B antigen, it's going to recognize the B antigen as being foreign, and it's going to produce antibodies. Antibodies are blood proteins that fight off anything that does not belong in the blood. Please write this down in your worksheet. So these antibodies are going to recognize the B antigen. They're going to bind to it, and then all of the red blood cells with the B antigen are going to go ahead and clump together. So this is a summary of the genotype, blood type, antigen, and antibodies. I want you to finish filling out this chart on your worksheet. So we're going to practice using Punnett squares with blood types. We're going to say that mom has blood type A and dad has blood type B. Start the Punnett squares, and we will notice that approximately 25% of the time we're going to have blood type AB. About 25% of the time, blood type A, and about 25% of the time it's just blood type B. And also blood type O occurs about 25% of the time in the offspring. I want to discuss blood types A, B, and blood types O a little bit more. So remember that people that have blood type O have antibodies for A and B, while people that have blood type AB do not have these antibodies. So when somebody has blood type O, they're able to donate to somebody that has blood type AB. But unfortunately, if somebody has blood type O, they're only able to receive blood from somebody else that has blood type O. So somebody that has blood type AB is considered a universal recipient, while somebody with blood type O is considered a universal donor. So this concludes our discussion on incomplete dominance and codominance.